Okay, we're looking at the answers for the investigating biology practice questions. This one here firstly is talking about different mice to determine whether or not the pituitary gland controlled growth. 90 mice were divided into 3 equal groups. Treatments and results were shown below. First, you've got to look at the table and try and interpret it as best you can. So it says that the mice were divided into groups. If we look at the groups here, they are listed across the top, 1, 2, 3. Now it makes sense that each group was given a different treatment, as it says here. So group 1 had their pituitary gland removed. So this is obviously the group that they're going to use to see whether the pituitary gland actually does anything. Group 2 had their pituitary glands removed but had daily injections of pituitary gland hormones. So obviously the pituitary gland must release a hormone, and they're seeing if simply replacing that hormone without the pituitary has any effect. Lastly, group 3 has no treatment whatsoever. So this is your control group, the one that you compare the other results to see if there's any change when you do this. If the pituitary gland has no effect whatsoever, then these results should match these results. Okay, so now they're looking at here, what have they measured? So that's our independent variable, what we've changed, plus a control independent variable here. So this is my independent variable control, so I can compare. These here then must be my dependent variables, which I measure to see if there's a difference. So what have I measured? My mass. That's my dependent variable at the start and at the end of the month. So at the start of the month, all these rats were around the 218 to 221 mark. So they're all just a bit over 200 grams in weight. Now remember, we're also trying to see if the pituitary gland controls growth. So you can assume that they're going to increase weight through that month if they're going to grow normally. After 1 month, we can see that these guys here have actually gone down in weight, these guys here have gone up in weight, and these guys here have gone up in weight. Now remember, this is the control group, the one that had nothing done to them. If they had nothing done to them and they increased weight, that is what normally would happen to a mouse with a pituitary gland. They have grown. So my assumption now is that these are obviously adolescent rats or baby rats, and they're looking at how well they grow. So it makes sense that they've increased weight. In fact, they've more than doubled it. In comparison, the ones with the pituitary hormone but no gland have doubled their weight as well. So they've done exactly the same as the control group. However, the ones where they have no pituitary gland and no hormone have actually lost weight. So it's safe to assume that the pituitary gland and its hormone that it produces most definitely do affect growth and weight gain. Because as soon as you remove the pituitary gland, that weight gain is reduced or even stopped. It's actually going backwards there. Plus, the only reason these guys here have increased weight, they had no pituitary gland, but the hormone that it makes has been replaced, and so they've actually shown normal growth. So we can even go further than this to say it's not the pituitary gland on its own that actually causes the growth, it's the hormone it makes that causes the growth. So here it says, which is the control group? Explain your results. Control group is group 3 because they're the ones which didn't have anything done to them, so we could compare. Number 2, what hypothesis does this data support? Justify your hypothesis by referring back to the data. Well, the hypothesis, or the educated guess that they had here, it has to be when the independent variable is changed. How? the dependent variable will then you make a prediction of will change and how that will change. So your hypothesis is always, when we change this independent variable in a certain direction, we think that the dependent variable will do this. So it's a guess that you're going to make for your experiment. What did we change? We changed the availability of the hormone in the pituitary gland. Okay, or in a very basic answer would be when the pituitary gland is removed, but we didn't just remove the pituitary gland, we added in the hormone here to test whether it's the hormone that the pituitary gland made that caused the change. So the guess would be when, now the educated guess that the data supports is that when you remove that hormone, that the growth will cease, or in this case even go down, because that's what they were testing. Does it affect growth? When the pituitary gland and its hormone are removed, organisms' growth will cease. And that is essentially the hypothesis that's been tested here. It has to include how they've changed their independent variable and how they've measured that to see what sort of changes occurred. Okay, so you must include those two for a proper hypothesis. When the pituitary gland and its hormone are removed, the organism's growth will cease, stop, be reduced. That's the sort of guess that they were testing, and of course their data does support that guess is correct. The next one is about vitamin C, and it says here that they're trying to see if it prevents colds. To test this, 10,000 volunteers were divided into 4 equal groups. Each person received a dose every evening for 1 year, and these were the results. Now they've said here, again, you've got to look for your groups and how they're changing them, because what they have changed is your independent variable. Okay, so there's your independent variable, the ways that they've changed it, and how have they measured it to see if there's an effect? Well, they've measured how many had colds. So this here is our dependent variable. Now, they said they had 4 groups, one with sugar, one with sugar plus vitamin C in different levels. And the question is, what's the control group? So think, what group has had nothing done to it so that you can compare and see if there's a difference? The answer is sugar. Now, this makes sense. You might say, why are they giving sugar to these guys? It's because the belief that you're being given a medicine can affect how you report your feelings and your wellness. So each of these guys were given a sugar pill. It was either a sugar pill with vitamin C's or a sugar pill on its own. So these guys didn't know whether or not they were getting given the vitamin C treatment or not. So the control group is the one that actually had nothing done to it, whether they knew it or not. And that's group 1, the control group, who only had sugar pills. They didn't have the treatment. And when you actually have a look at it, group 1 had 20 colds in the year. Group 2 and 3 had almost the same, so no real difference at all. Group 4 had 10 colds, so that actually was a reduction of half. When you go to group 10, the number of colds halved. So you could actually assume that if you give at least 9 grams of vitamin C per day to a person, 1 and 3 are useless, at least 9 grams of vitamin C per day, that it will reduce your chance of getting a cold. Next one. A test was carried out to assess the fertilizer requirements for a 1,400 hectare wheat and soy farm. Different types of fertilizer were applied to 4 1-hectare plots of land, as shown below. So each of these hectare plots were given 1 ton of urea, 1 ton of superphosphate, 1 ton of animal manure, and no fertilizer at all. So first of all, where's your control group? Because all of these are your independent variables. These are the things that you've changed. So they're the type of treatment is what your independent variables are, different treatments. Your control variable is this one here. Not control variable. Remember, control variables are all the little things that you keep the same to make it a fair test. Control group is what this is. A control group is a group that has no treatment, no change, so that you can compare it to the ones that have had changes. Control group, not control variable. Control variables are different. Now let's see if we can look at the results. So here's the different independent variables over here, and there's my control group there. And here are my dependent variables, or my measurements here. So the wheat was then planted and the following yields were harvested. Urea was, let's look at the control group first. No fertilizer had a 1.0 chance, so a 1 ton yield per hectare. Animal manure and superphosphate had 1.5 and urea had 2. So what did this test demonstrate? Well, compared to the control group, so this is the one where they did nothing to the soil, did these treatments have an effect? Yes, all 3 of them did improve the growth. Okay, so all three treatments worked. All 3 treatments increased the yield, but urea was the best because it increased it by doubling it. Explain your reasoning for the response in 8a. What's that? What has this demonstrated? Well, in other words, explain it, relating it back to data. Okay, so comparing to the control group, manure and superphosphate increased it by 50%, from 1 to 1.5, and urea increased it by double. Okay, next question. The table below shows plant and animal counts for the years 1985 to 1990 in a small area. The count includes all of the plants in the area. Okay, so these are all the plants and all the animals in a specific area, how many they have of each. As you can see here, now when you read things and it has a timeline, you've got to start telling the story of what the graph or the table is giving you here. So this is telling me that over the years from 85 to 90, this plant was really low. It went up, went down, died. Okay, so you can sort of do a little pattern here of what happened to this plant and think about it as you tell the story. Plant 2, it didn't exist in the first 2 years, and then it went up. So it basically had nothing, and then it went slowly up. Plant 3, it went down. Saved me drawing lines over my graph. I'm just going to do this. Plant 4 went down. So we've got this plant here that went up, then down. This plant here that only went up. These 2 went down. This last plant here went up and then down. So there it went down. Animal A. He was present at the beginning and then he died off. Okay. Animal B was present in high numbers at the beginning and then started to go down but then up. Animal C was not present for the first 3 years, then went up and then down. So it says here, state the most probable food for animal C and justify your response. So here's animal C. It wasn't even present in the first few years. It went up and then down. Now, if it's relying on a certain plant, its population numbers will follow the same pattern that its food source does. So let's have a look which plant has the same pattern of up and then down. Well, most definitely this one does here. These plants weren't in existence here at all, so they couldn't actually help animal C. This plant here was slowly rising up, and this plant here was going down. So considering that animal C had a definite increase and then a decrease from 88 to 90, the only other plant that shows that same pattern that animal C follows is plant 5. So shows the same growth and shows the same population growth and decline. Okay, next one. The graph below displays seed germination of 2 acacia species in response to heat treatment. The control group was not given any heat treatment at all. So here we've got acacia species. Does heat treatment affect, well, by the look of this, seed germination? Because what are they changing? The independent variable is heat, whether they expose seeds to heat. And what are they measuring? They're measuring seed germination. So it's safe to assume that they're trying to see if heat affects seed germination. So this is why you must always look at what are they changing, what are they measuring. That leads you straight to what they're trying to investigate. What did they change? Heat. What did they measure? Seed germination. So let's see if heat affects seed germination. This says along here for these 2 acacias, each one's different, the number of germinating seeds, and along the bottom here, this is the duration of heat treatment. So they're actually seeing not just if they need heat, but if they need a little heat or a lot of heat to allow them to germinate. And of course the control group was given none. So your control group here is this dark one here. So if we have a look along here, there is no control group showing much germination at all, except for maybe a little bit here. So the control group for this acacia is not germinating much, even when you do give it heat treatment. It showed only minimal germination because it hadn't been given any heat. So basically the control group in this one didn't really germinate much. Now let's have a look at the others. Now on top of this, they haven't just exposed it to heat, they've exposed it to different temperatures of heat. So this is 90, then 120, then 150 degrees Celsius. So there's 90, 120, 150, 90, 120, 150, and over for different periods of time. So for just 2 minutes of exposure, the 150 degrees Celsius germinated the most. For 10 minutes of exposure, even the lower temperatures germinated better, but again, the 150 degrees Celsius germinated the most. 30 minutes exposure, they actually didn't germinate as much as the 10 minutes. This one in here, that 120 degree there, it's the same. Whether it was 10 minutes or 30 minutes, it hasn't really increased at all. It's increased a bit, but not much. 3 hours, that 120 degrees Celsius started germinating really well, and the 90 degree one increased as well from there to there. So it's looking at here, the lower temperatures, you need to really expose them for a longer period of time for them to cause germination. Now look at this, the 150 degrees Celsius, which was so good over here, there wasn't much germination at 30 minutes, and at 3 hours they had nil, which means basically it's killed off the seed completely and it's not going to germinate. So 150 degrees is fine for a short period of time, but then if you continue exposure for a lot longer, say 30 minutes, it starts to kill off the seeds. By 3 hours, 150 degrees will completely kill the seeds and there will be no germination. So when you think about it, it's either high temps for a very short amount of time is good, or lower temps for a longer period of time is good for the seed germination of this little guy here. So it's either 150 degrees Celsius for short periods of time equals germination, but over 10 minutes, and that little guy there will start to, well, in this case, less germination and then eventually no germination. 90 and 120 degrees, you can see here, not good germination until you get, well, with this one here, even more than 30. You need to have about 3 hours before it will actually germinate well. Same with this one here, 3 hours before it will germinate well, and 6 hours, even better for the 90 degrees, but at 6 hours the 120 degrees started to die off. So for 120 degrees, it has its best growth after 3 hours exposure, and then it will die off. And then 90 degrees, it needed at least 6 hours to have its maximum germination. And I'm assuming that too much more will actually kill it off as well, but we haven't got that data. So the higher the temperature, the shorter amount of time it needs to be exposed for it to germinate. And at those higher temperatures, after a while, it seems to kill off the seeds. Now let's have a look at this other acacia here. Same thing. But first look at the control group. All of them germinated down here. All of them germinated. So when you look at the heat exposure here, for say this is the 90 degree one, the 90 degree didn't actually have much better germination than the control group. Neither did the 120. And the 150, well, they had less germination than the control group, so it actually damaged the seeds. 10 minutes here, if you look at the 90 degree one, there actually isn't any difference. So it looks like exposing this little guy to heat is actually not helping it germinate any better than it would on its own. Notice that the 120 and 150 ones here, these are almost dead here and completely dead over here. So these high heats are not good for this little acacia, and the 90 degree heat doesn't seem to have any difference compared to the control. So the question is, did it really need to be heated up to germinate? No, it didn't. Whereas this one, the control group, there was almost no germination on its own compared to the ones that were exposed to heat. So we have two different acacias that respond to heat treatments differently here. Let's have a look at the question. Interpret the data and draw a conclusion. Well, basically, the first acacia definitely does respond to heat, but higher heats will result in good germination, but prolonged exposure to high heat will kill them off. So it's either high heat for a very short period of time or lower heats for a longer period of time, or you will kill off the plants. This other acacia here, it actually didn't show any significant change compared to the control group, so heat isn't required for germination. However, high heat exposure will actually kill off the seedlings and they won't germinate at all. So there's definitely a difference between those two acacias in how they respond to heat. Okay, next one. The results below were collected in an investigation on the effect of temperature on the reaction rate of an enzyme. So the independent variable that we're changing is temperature, and what are we measuring? The reaction rate, how quickly it makes a product. Now if you have a look here, as the temperature increased, you can see here we went from 1 to 2 to 3 to 4 and then down again. So there's actually a pattern here like this. So when you look at that temperature at first most definitely does increase the reaction rate until we get to 40 degrees, and then the reaction rate actually goes down and gets to 0 by 60. So what conclusion can be drawn from these results? Explain why this occurs by linking it to enzyme theory. Basically, temperature does increase. Temperature does increase reaction rate up to 40 degrees Celsius, and then the reaction rate declines to 0 by 60 degrees. Theoretically, this does hold true. When you think about it, the maximum temperature our body can actually handle is around 40 degrees Celsius, and after that we know that enzymes start to denature or fall apart. So after 40 degrees Celsius, the enzymes are actually exposed to temperatures that interfere with their ability to function because it affects their structure, which then affects their function. By 60 degrees Celsius, there is no enzyme activity, so we can assume that that enzyme is completely denatured by that point and therefore not able to function at all.